We now turn our attention to the hereditary topics, so genetics. We need to start by going through a whole list of various definitions, and it is important that you know the meaning of all these terms so you can apply these things to the questions when you're asked. So starting with an allele is a form of a gene, such as having blue eyes or brown eyes. They're both alleles for eye colour. They're given by letters, so blue eyes being a recessive trait will have a lowercase b denoting it. Brown eyes being a dominant trait will have a large uppercase B denoting it. A gene is a section of DNA which codes for a particular protein. The genotype is the alleles belonging to an organism. You'll have two alleles, so if you have brown eyes, for example, you could be uppercase B, uppercase B. Blue eyes would be lowercase b, but that is your genotype, whereas your phenotype is the physical appearance of a trait, so that is whether you have brown eyes or blue eyes. It is not the same as the genotype. Make sure you're clear on that, and we will look at some past paper questions to help you here. Homozygous means that you have two of the same alleles, such as two lowercase b's or two uppercase b's. Heterozygous means that you have two different alleles, such as a capital B and a lowercase b. A recessive trait, the alleles are only expressed in the homozygous state. That means you'd need lowercase b. As soon as that uppercase B is present, then the dominant trait would be expressed. Dominant is when only one copy of an allele is needed to express a particular trait. This is different to codominance, where both alleles are present in the phenotype. A good example here is the snapdragon plant. If you have a red snapdragon flower, a white snapdragon flower, rather than one colour being dominant over the other, what would happen is that both alleles would contribute to the phenotype, and in this case you'd see a pink flower. Moving on to haploid and diploid. Haploid means containing only one set of chromosomes. In humans this is 23 chromosomes. Diploid means containing two sets of chromosomes. In humans this is 46. Monohybrid inheritance means that one gene is responsible for a particular phenotype. Polygenic inheritance means that many genes are responsible for a phenotype, and in actuality that does tend to be the case for most traits. In most cases, many genes contribute to a particular phenotype. So let's take a simple genetic cross. A heterozygous brown-eyed mother and homozygous blue-eyed father want a baby. What is the probability that their baby will have blue eyes? So we're going to use the following layout in order to make sure we answer this properly. Please don't rush this. You want to write mother, father, phenotype, genotype, gametes. So this is the preparation you do in order to make sure that you get your Punnett square nice and accurate. So set yourself up like this. You should have plenty of space in the exam to do this. Now look at the clues given in the question. We're going to use B to represent eye colour. So we know that we have a heterozygous brown-eyed mother. The fact that it's heterozygous means that the genotype must contain one uppercase B and one lowercase b. We know that the father is homozygous and blue-eyed. Blue, remember, is a recessive trait. Homozygous means that those alleles are the same. So there's the father's genotype. We're now ready to write down the answers in our table. So the phenotype of the mother is that she has brown eyes, because remember, phenotype is the physical appearance. The father has blue eyes. Now list the genotype. We know the mother, I've already worked it out, is capital B, lowercase b. Father is lowercase b, lowercase b. The gametes, this means the mother's sex cells, so this is the egg cells. Her egg cells will either have a big B allele or a lowercase b allele. The father's sperm are all going to be that lowercase allele. This means you're now ready to set up your Punnett square. So it should look something like this. Just list those alleles again. It's up to you if you circle them. Give yourself plenty of space. And the Punnett square will enable you to work out the possible likelihood of that baby having blue eyes. So just list the various genotypes, and you should give yourself space to list the phenotypes underneath. So this baby here will be brown-eyed, this one will be blue-eyed, brown-eyed, blue eyes. So in terms of answering the question, our probability is that there's a 0.5 chance of the baby having blue eyes. Although percentages aren't technically probability, you'd also get away in biology with writing 50% chance or 1 in 2 chance. But it's important that you do this because these questions tend to be worth around 4 marks, and that will enable you to get the marks for linking the various phenotypes and genotypes at the end. So I do implore that you spend the time writing this out properly. Let's take an example now involving cystic fibrosis. So we have a carrier mother and a carrier father of cystic fibrosis. Cystic fibrosis is a nasty disease caused by the recessive allele. I'm going to call that C. So in order to have cystic fibrosis, you need 2 lowercase c's. So the carrier mother automatically tells me that her genotype must look like this, the same for the father. It's because they contain that lowercase allele, which is responsible for causing cystic fibrosis, but because it's a recessive disease, they've got this healthy allele, which luckily for the carriers means they don't suffer with any of the effects. But if we actually have a look at the cross this time, we're going to use the exact same layout as before: mother, father, phenotype, genotype, gametes. Like I said, it works every time. So you can write healthy or carrier here, it's up to you. I've already worked out the genotypes. So half of those egg cells will be healthy, half will contain that lowercase c, same for the sperm. And then if we look at the Punnett square, that's a healthy child. Here is a carrier, still healthy though. It's that final child here which will have cystic fibrosis. We can say that these ones here are carriers and that this one's entirely healthy. So it's 25% chance or 0.25 chance of having cystic fibrosis. I've already said that a diploid number of chromosomes means 23 pairs of chromosomes. 1 pair is actually known as the sex chromosomes, and this determines whether someone is male or female. Males have an XY sex chromosome combination, females have an XX chromosome combination, and it's possible to do a genetic cross to show this. Let's now look at sex inheritance. So we know that the mother is going to be female, the father is male. I've already said that the sex chromosomes in females is XX, in males it's XY. So that means all of the female's eggs will contain an X. Half of the male sperm will be female, the other half will be male because they'll contain a Y. And again, you're ready to get going. So this baby here will be female, this baby here will be female, this baby here will be male, this baby here will be male, hence why there is a 50-50 chance of having a girl versus a boy. Pedigree analysis is used to show how a particular genetic condition is passed on from generation to generation. They are in effect family trees, so you will have the grandparent level, the parent level, the grandchild level, and you'll be able to see a male and a female and the children they produce, and then using the key you'll understand whether they have a particular condition or not. The great thing about pedigree analysis or pedigree tree is it allows you to work out whether a particular condition is a recessive or dominant condition. So for example, if 2 healthy parents, and you'll be able to use the key to work out if they're healthy or not, have a child with the condition, it tells you that the condition is recessive and that both parents must have been carriers. That's the only thing that can be true if they're both healthy but their child has the condition. Equally, you'll be able to work out based on the number of people suffering from a particular disorder whether it's a dominant condition or not based on how many people have it in that pedigree tree. And we'll look at an example now to really take you through step by step. So you can work out the sort of thinking you'll need to unpick these questions. Familial hypercholesterolemia, FH, is an inherited condition caused by a dominant allele. People with the condition have high levels of cholesterol in their blood, and this increases the risk of dying from blocked arteries. The diagram shows the pattern of inheritance in several generations of a family with FH. So as I said, here's the all-important key down here that will tell you that all the squares are males, all the circles are females. If it's shaded in, then they must have the disease. If it's blank, then they are healthy. So the first thing you want to do is work out what genotypes would be required to cause FH. We know it's a dominant condition, which means that either this combination or this combination will cause the disease FH. The only way you can be healthy is to have the recessive genotype, which looks like this. It's very important that you get to grips with what the information is telling you. So if we look at part A, person A is heterozygous for FH. Use this information to complete the table. So we've been told that this person has 2 different alleles. We can immediately label all of the unaffected healthy people's genotypes because I've already said they must be lowercase h, lowercase h. So we're being asked how many people are homozygous recessive. Homozygous means they have 2 of the same allele. Recessive tells me that they're both lowercase. So then it's simply a matter of just counting up all of the homozygous recessive people. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. What about homozygous dominant? Homozygous, 2 of the same allele. Dominant tells me that they're both capital H. So now we need to work this out. This daughter here has the disease. We know that she'll have only got a lowercase h from her mum. What about this son over here? Same setup. He has the disease, so he must be uppercase, but he's got that lowercase h from his mum, and you just keep working it down. Look, it's the same combination here, so I can immediately write that allele combination, that genotype, then working down to person N. They'll have inherited the lowercase h from their mum, the uppercase h from their dad in order to have the disease. The same is true for all 3 offspring. So actually, if you sit back and look, how many people have this genotype? Well, the answer here is 0. Person G and person H have 3 children who all have FH. What is the probability of G and H having 3 children who all have FH? So G and H are here. Let's do a super quick Punnett square to work out the answer to this. So these 2 people here will be diseased, so 0.5 chance of having the disease because they have this genotype. So in order to work out the chance of them all having FH, because it's probability in maths, remember if it's person 1 and person 2 and person 3, you need to multiply that probability 3 times to get 0.125 as your answer. Some disorders are sex-linked, which means that the allele for the particular trait is located on the sex chromosome, so that would be the XX chromosomes in females, the XY chromosomes in males. And you often find that with sex-linked characteristics, they're more common in one sex compared with another. For example, red-green colour blindness is way more common in men compared with women. Most sex-linked diseases are actually located on the X chromosome. The reason for that is because the Y chromosome is much shorter and therefore has comparatively fewer genes located on it. Just think about the length of an X and a Y in the alphabet and you'll see that the X is longer. X-linked dominant traits are more common in females, and there's quite an obvious reason for this. It's because they have 2 X chromosomes, which means they have 2 alleles. So either allele may be dominant and cause disease, so it doesn't matter if it's homozygous or heterozygous. If it's dominant, it will cause the disease in the female. Whereas X-linked recessive traits are much more common in males. The reason for that is they have only 1 X chromosome, and therefore there's no potential for masking by another allele because quite simply it doesn't exist on the Y chromosome. If they have that copy, even if it's that lowercase allele, the recessive trait, the fact that they don't have a dominant allele on another X chromosome to mask that means that they'll suffer from the disease. So we can make the following conclusions: that for sex-linked diseases, it's only possible for females to be carriers. Males always inherit their X-linked allele from their mother. and females cannot inherit their X-linked allele from an unaffected father. Red-green colour blindness is a recessive sex-linked disorder. The allele for colour blindness is located on the X chromosome. A heterozygous female has a child with a normal-sighted male. What are the possible genotypes and phenotypes of the offspring? So we're going to go female, male. This setup works for all questions. Phenotype, genotype, gametes. So we've been told that red-green colour blindness is recessive. So in order to have the disease for the female, they need to have this allele combination, this genotype. So we know based on the fact that they have this uppercase that they must have normal vision. We've been told that the male is normal sighted, so I can write that here. The genotype they've already given to us. What are the gametes? So we'll just separate those nice and carefully. Now we can draw a Punnett square. So now we're ready to list our possible outcomes for these offspring. Okay, so in terms of, we're going to have a female here with normal vision. Here's a male with normal vision because they have that uppercase C. Here's a female, normal vision. Technically this one's a carrier. But then this one down here is a male with colour blindness because they have that lowercase c responsible for the red-green colour blindness. So in terms of outcomes, it's 0.25 chance of being a male that's colourblind. We're now going to move on to discuss haemophilia. Now, haemophilia is a blood clotting disorder. It's caused by a recessive allele. That recessive allele is located on the X chromosome only, so it is a sex-linked disease because it's located on the X chromosome. So if we consider various genotypes, there's a female that will be healthy. Here's a female that again will be healthier but a carrier due to the presence of that allele there, which remember is the recessive allele responsible for haemophilia. Here's a male that will be healthy. And here's a male that will have the disease. And that's due to this lowercase h here. And remember, we said that the allele for haemophilia is only located on the X chromosome, so that's how I can infer all these things. Now let's look at a Punnett square cross. Haemophilia is a blood clotting disorder caused by a recessive allele found only on the X chromosome. A man needs to inherit only one allele to have the disease. What is the probability of a male haemophiliac and a female carrier having a haemophilic child? So, ready to go again. So male has the disease, female is healthy. We can infer that from the wording of the question. The genotype, so here is the sex chromosomes for the man. We know that he's a haemophiliac, which means he must have that lowercase h in order to have the disease. Remember that allele will only be located on the X chromosome. The female, we've been told, is a carrier. There's her female sex chromosomes. For her to be a carrier, she'll need that genotype. So we've done all the hard work. Let's do the Punnett square now. So this is a female healthy carrier. This is a male who's healthy. This is a female with the disease. And this is a male with the disease. Huntington's chorea is a dominant genetic condition that results in the nervous system not working correctly. It is not a sex-linked condition. Diagram 1 shows a family pedigree. Some members of the family have the condition. Remember to use that key so we can see straight away. Here we have a healthy male, a diseased female, healthy male, female, healthy male, diseased female, healthy male, diseased male. Evaluate the evidence in the pedigree that supports the statement that this is not a sex-linked condition. It's because if you consider the individuals which are affected, 3 of them are female, 2 of them are male, so we can make the point first of all, it's not a sex-linked condition because it appears in both males and females in similar numbers. Evaluate the evidence in the pedigree to support the statement that this is a dominant condition. So dominant means that you would expect to see lots of sufferers, and indeed you see people suffering in every generation. You could even work out some of the genotypes. So we'll use H to represent. That woman there must be large H, lowercase h. The reason I know that is because her partner here is definitely lowercase, because he doesn't have the disease. And if you look at their children, we can see that this male here doesn't have the disease, this female does, this male doesn't, this male does. So based on that, we know therefore that she must have passed on her capital H, so we can see that the disease is present in heterozygotes because she is a heterozygote, and that only one allele is needed for a person to be affected by this awful disease. So we write here that the condition is present in the heterozygote. So only one allele is needed. And some children from affected and unaffected parents, as in the case of that first generation, are affected whilst some are not. A man has the same genotype as person 1 in the pedigree in diagram 1. Let's have a look at person 1. It's that person down here. We know they're unaffected, which means there must be lowercase h. Just making notes to myself. A woman has the same genotype as person 3. Let's look at person 3. Because person 3 is healthy, they must also be lowercase h. These two people are expecting a child. Calculate the probability that the child would be a boy, not showing the Huntington's condition, giving reasons for your answers. So we just need to draw out everything I was doing before. So we have a mother and a father. Phenotype. This will become all our reasons by laying it out like this. So we know they're healthy. We know their genotypes are this, which means their gametes must be this. We don't even really need to do a Punnett square because we can see that there'll be 100% this, which is healthy, which in probability terms means a 1.0. And then this thing about having a boy, remember there's a 0.5 chance of having a boy, 0.5 chance of having a girl. So because it's probability and you need both to be true, that they're healthy and a boy, you multiply the two together based on maths probability, and the answer here is 0.5. A man has the same genotype as person 2. Right, so we are looking at this person down here. We need to work backwards. We know that this man up here must be lowercase h. This girl down here must be lowercase h, which means this woman here must be capital H, lowercase h. in order to pass on the healthy allele to this girl, but the unhealthy allele to this boy. So I can immediately discern that that is number 2's genotype, so we'll steal that. And then what about the woman that has the same genotype as person 3? I've already worked that out, it's lowercase h, lowercase h. Explain the possible phenotypes and genotypes with respect to the Huntington's condition of any offspring they produce. So again, we can use our layout. So we're going to have man, woman, phenotype, genotype, gametes. So we know that this person here has the disease. This person is healthy. Sort out your gametes. Draw your Punnett square. So there's offspring 1. So we can see that there's a 50% chance of being healthy. Make sure you list that genotype. 50% chance of having the disease.